Hi everyone, welcome back to discuss on formulation development, process parameters and risk assessment is going to be discussed as a part of this module. There are previous models we have discussed pre-formulation, parameters, what are the important studies to be done. So now in this model, we can understand certain formulation activities and a QBD based approach to be applied as a part of formulation development. And there will be four models have been generated to cover a different type of formulation and its process parameter and risk assessment. Formulation development, if you look at, we have tabulated here, there are multiple dosages. One is a solid oral, we all know that it's capsules, tablets, and pellets, and other powder stuff also. At certain stage, the granules and the fine powders. In terms of the sterile dosage forms, injectables, which is nasal solutions and lyophilized products. And liquid, syrups, elixir, gargle, enema, and other suspensions. Semi-solid dosage forms, ointments, creams, paste, and other components. Gases, aerosols, inhalers. We will not look at all the dosage forms. We can take certain examples and we'll look at how this development would happen in Indian pharmaceutical industries. What are the approaches they follow when they develop the formulations during part of development and scale up. First of all, when we start a formulation development, a formulation scientist should aware what is QBD, which is nothing but quality by design, which in recent days FDA started asking any kind of formulations should have been developed with this approach. QBD is nothing but you have to assess the risk and the robustness of the formulations what you have designed it. QBD facilitates design of products and processes that enhances the product's quality, efficacy, safety in the interest of patients. The QBD is a systematic process to generate a robust process with the help of quality risk management. And also, it emphasizes the product and process understanding and process control based on sound science and quality risk management. So this is a definition of the QBD. We can look at what all the elements are involved as a part of QBD. QBD has all the variables, input materials, a process, and critical process parameter which we have to be identified, and that needs to be adjusted by measurements of CQA, which is nothing but we call it critical quality attributes. And all these inputs could give you your final product. So the variables need to be optimized by using a quality based design. For example, if you have a material having three different grades, those all three different grades need to be studied as a part of quality based design. Are we getting any kind of a difference in terms of critical quality attributes by using a different grades of material grades? So one of the important goals of QBD is to ensure that all the sources of variability affecting a process are identified, explained, managed by appropriate measures. In pharmaceutical industries, there are terms is being used as a part of QBD. One is QTPP, the second one is CQA, and third one is CMA, and the fourth one is a CPP, followed by FMEA. Maybe we can discuss in detail on each forms what we have defined here. QTPP is nothing but a quality target product profile as it relates to quality, safety, and efficacy, considering the route of administration, dosage form, bioavailability, strength, and stability. CQAs, it is nothing but critical quality attributes of the drug product, so that those product characteristics having an impact on product quality can be studied and controlled. Determining the critical quality attributes of the drug substance, excipients, etc. Selecting the type and amount of excipients to deliver drug product of the desired quality. Selecting an appropriate manufacturing process, defining a control strategy. So when we start drafting a QTPP, a formulation scientist should know what is your target product profile in terms of your dosage and in terms of your administration and your therapeutic index and certain microbiological attributes in certain cases. When we talk about the critical quality attributes, which is nothing but a testing tool, or what all the critical quality parameters which can use to measure the quality of the product. For example, you can say it's a degradation, you can say pH, you can say powder properties, flow properties. There are multiple factors where you can list it down, and from those factors you can categorize it. The level of risk shall be defined for the critical quality attributes. Similarly, we call it a factor which is called a CMA, which is nothing but critical quality material attributes. The critical material attributes talks about each material and its variability contributes to the final product. For example, a lactose. Lactose is being marketed in the market with a different grades and different particle size and different moisture content. Now, as a part of CMA, a formulation scientist should study the effect of all the parameters, whether it is an LOD, It's a particle size, because these all parameters will directly impact the final product quality. Example, a particle size, it is not appropriately selected, the lactose particle size, in comparison with your drug substance, where we will be ending up with certain improper flow issues during tablet compression. And followed by CPP, which is nothing but critical process parameter. The critical process parameter talks about what all the manufacturing process is critical while you formulating a product. For example, if you make it a dry granulation, a dry granulation requires a granulator or a sifter which can define the particle size. A dry granulator equipment needs a lot of parameter, which is nothing but your feed rate and your screen size and your roller speed. These are the main key parameters to be identified and should be optimized with a defined ranges, which can give you a final product quality as per CQA. And followed by, once you complete all these activities, QTPP, CQA, CMA, CPP. Finally, you will be ending up with making a kind of failure mode effective analysis, which is nothing but FMEA. The FMEA would give us an overview of the complete product development. Each stage will be defined and that will be discussed well, and that has to be categorized with respect to appropriate risk number, which globally recognized by most of the organizations. Let us see in future slides. We have given certain examples for your understanding, how that is going to be help you while you start your career in pharmaceutical industry. This is an example how this QBD aspects, as we defined it, you can see the flow chart, a target product profiles, a critical quality attributes, a risk assessment, a design space, control strategy, and followed by a life cycle management. QTPP, we have discussed well. A QTPP is a prospective summary of the quality characteristics of a drug product that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of the drug product. That's the definitions. But we have discussed what is the importance of QTPP, why we are doing this, because it is an essential element as a part of QBD approach. In most of the ANDAs, the target profile should be defined based on the characterization of an RLD. So RLD is nothing but a reference listed drug which shall be characterized, understand, the basic information, whether the formulation is a tablets, and what is the size of the tablets, what is the weight of the tablets, what is the dosage of the tablets, what kind of strength, and what is the packaging, how it was done, the secondary packaging, is there any product needs any maximum daily dose. So these all information shall be gathered and that should be defined as a part of quality target product profile. It also includes a product attributes that needed to ensure equivalent safety and efficacy to the RLD. Here we are presenting one example for your understanding as a case study. You'll get many more in a public domain, and you can also refer it the ICH guidelines. So this is a typical QTPP which normally follows in most of the industries, which talks about dosage form, dosage design, route of administration, strength, pharmacokinetics, stability, and the drug product quality attributes. If you look at the drug product quality attributes, all the test parameters which is importance for the tablet dosage has been defined here. The container closure system, administrations, and the target to be defined it based on our understanding, oral and 20 mg tablets, a similar food effect of an RLD when you talk about administrations or concurrence with the labeling. And also each parameter should be justified with our understanding why that the parameters have been selected, how it is important to us to consider as a QTPP. Similarly, a CQA also has to be defined based on the physical, chemical, biological, or microbiological properties. We understand that a physical parameters, kind of a description, a pH, or appearance, a certain level of a reconstitution, solution appearance, and chemical when we talk about the degradations, assay, a biological, something there are certain products need the biological reactivity studies or certain kind of BE study has to be done which proves that equivalency, and microbiological, a certain product need a control on microbial load on the product. For example, if you look at an injectable, you need bacterial endotoxin should be controlled and your aerobic and anaerobic levels should be controlled. So with this understanding, you can define a CQA. Normally the CQA, it varies with respect to the dosage. For example, a tablets would list down what are the critical quality attributes. Similarly, the parenteral, which varies with some special test, kind of a liquid appearances, it varies, microbial attributes, it varies, and pH commonly is being used in parenteral dosage, whereas in tablets, it is not. So this is a variability you could see, and similarly, when you talk about a semi-solid dosage, you can see the rheological properties of the semi-solid. It's very, very important factors. So these are process parameters or the critical attributes to be listed down, and that needs to be discussed as similar to a QTPP. Here we have presented a few commonly used CQAs for each dosage. For example, tablets, injectables, and suspensions with the tablets. You can see a friability, content uniformity, dissolutions, and microbial testing. And similarly, injectable dosage, bacterial endotoxins, bioburden, color, and clarity. Suspensions, assay per droplet, particle size distribution, viscosity, spray droplet size distributions. Accordingly, for the dermatological product, you can see the rheological properties, appearance, phase separations. So these are all the very key CQAs is commonly is to be defined for any product development. Here we have just tabulated an example for your understanding. See, for this product, the appearance, a color and shape acceptable to the patient. No visual tablet defects are observed. Is it CQA? No, it's not the CQA. So here the justification has been done. Why it is not the CQA? The color, shape and appearance are not directly linked to the safety and efficacy. Therefore, they are not critical. The target is to set to ensure patient acceptability. Then what is CQA? Can we go? Yes. You can see a content uniformity, which is nothing but CU, which always performs as per USP 905. The question here, is this a CQA? The answer is yes. Why it is CQA? Variability in content uniformity will affect safety and efficacy. Both formulations, process variables impact content uniformity. So this CQA will be evaluated throughout product and process development. The way content uniformity get alters based on the process, based on the critical material attributes. So it is very important to study this content uniformity to understand the effect of CMA and CPP. Similarly, there are assay, dissolution, identification, degradation products, residual solvents, water content, microbial limits, all considered as a part of CQA. Of course, there are few parameters which can be justified. It is not affecting the product quality. Hence, you can categorize it as not a part of or not a risk parameter for the particular formulation. CQA, you can categorize it based on your understanding with a prior knowledge on the certain products, where you can categorize it as a 3 levels, which is low, medium and high. Low, broadly acceptable risk, no further investigation is needed. Of course, the medium is acceptable, but investigation is needed. But when you talk about high, which is unacceptable, a further investigation needed to reduce the risk. Or what kind of development or studies to be done to understand this high and to minimize this high risk when we take the formulations for the development or taking into the scale-up level. Here is an example how it is being categorized. If you look at a drug product CQAs categorized against one drug substance attributes, considering a drug substance how it is impacted. If you look at the CQA which is assay, of course it gets changes with respect to the particle size distribution and the chemical stability. We know that the degradation happens, then automatically the impurities get increases and your drug content may go down. This would be the one of the factor. And similarly, if we talk about content uniformity, If there is a change in the particle size distribution of a drug substance, the content uniformity of the drug product get changes, and also the flow properties also. Similarly, a dissolution. The dissolution get alters or differs when the solid state forms, because each polymorphs have a unique solubility. So accordingly, this dissolution get changes. Of course, the particle size distribution, solubility as well, and also the degradation. Similarly, if the solid state form changes, the degradation also, because each polymorphs behave differently based on the route of synthesis. Of course, the chemical stability also. Here, we have given certain initial risk assessment of a drug substance attributes. Of course, we have discussed in a previous slide on each parameters. In this case, for example, we can take it a solid state form of drug substance attributes. What all the parameters are affecting on drug product CQAs? Which is nothing but assay, content uniformity, dissolution, degradation products. Why? Justification, you can look at that why it is affecting. A drug substance solid state form does not affect tablet assay and CU. So hence the risk they have categorized is low. But whereas a dissolution, a different polymorphs of the drug substance have different solubility and can impact tablet dissolution. Hence this is considered the risk is high. Similarly, the drug degradation products. Drug substance with the different polymorphic forms may have different chemical stability and may impact the degradation products of the tablet. Hence, the risk is medium. So you can go through in detail the tabulated given examples on each parameters, how it is being categorized, how it is linked with the CQA, and what is the justification. So what are the stages are involved when we identify a process variables for further study? One is identification of product CQA, and each process step identify intermediate CQA that impact drug product CQA. And step 3, identify material attributes and process parameters that may impact intermediate CQA of the process step. This we have discussed in detail the previous slides. Here we can see how these all interlinked. A case study has been tabulated and we have put it in the flowchart for you for your understanding. So the first column is materials, and this is a process. The process involves blending, dry granulation, milling, lubrications, compressions. And as an output, what all the output we are going to get it? What kind of controls we are going to have it? And in-process controls. This defines at each stage what kind of controls we can have it to understand a product quality. And along with that, we can also define an associated CQA, which is nothing but critical quality attributes. Of course, the CMA is part of this materials. The CMA should be considered for each component to be defined it on its variable, and that need to be linked with a process and as well as a CQA. If you look at when you mix all this component, the blending should be involved, which is nothing but a critical process parameter. So there will be a check to understand a uniformity, which is nothing but a CQA, and that to be tested as per USP, and you can have a control on the dose uniformity. Similarly, you can also see the dry granulation where you can see the density and the associated CQA would be dissolution. So these all the parameters are very well interlinked, and these all the flowchart should be defined for every formulation irrespective of dosage units, whether it is tablets, capsules or injectables or dermatological product. Here we have put it how the CQAs are linked with the process steps. Let's take an example. For example, it is an assay. How it is impacting. A pre-roller compactions, blending and lubrications. Will that impact? Yes, it is impact on medium. And milling, yes, it impacts based on the size on the milling and your particle size and screen size, the milling would happen. This would impact the uniformity and as well as your assay. That's the reason the content uniformity we have categorized all these are high except final blending. Of course, these all process parameters definitely would not degrade the product, hence these are categorized as low. Here, as an alternate example to understand a liquid formulation which is nano suspension. Here, if you look at the left column which is critical quality attributes, the right one, the compounding, viscosity modifiers phase, tonicity phase, EPA phase and preservative phase. These all are clearly interlinked and it has to be categorized at what level the risks are. For example, if you look at assay per spray, which is very critical parameter, it gets changes with respect to your viscosity of the formulation and with respect to your APA also, because there will be a lot of changes happen when we talk about the phasing mixing time. The phase mixing time is very important factor when we formulation a nano suspension. Of course, the drug assay also. Viscosity is one of the major CQA for formulating any kind of suspensions because your particle size may get changes or your uniformity would get alters if you have not get an uniform or homogeneous suspension. Coming to the critical material attributes, we have discussed the interlinked CQAs with the CMA. So now the definition says what it is: a material or process whose variability has an impact, a critical quality attribute, and therefore it should be monitored or controlled to ensure desired drug product quality. So now we have looked at it. What are the critical attributes, material attributes for each excipients and the drug substances, how it is impacting drug product quality, and as well as critical quality attributes. Here we have tabulated a drug product CQA, how it is impacting a drug substance. This is an additional example which we have defined it. Take an example of dissolution of drug product CQA, how it is getting impacted by your drug substance attributes. If the solid state form changes, it is high, and the particle size distribution, it is high, and solubility also is high. Based upon the physical, chemical, and biological properties of the drug substance, the initial risk assessment of drug substance attributes on drug product CQA is defined well. Similarly, you guys can tabulate it for individual products based on your understanding, and each CQA shall be very well discussed, and that shall be categorized against drug substance attributes. Once you have done this QTPP, CQA, CMA, and critical process parameters, you can have a control strategy in place for each factor. For example, I'm taking an example of grade of lactose monohydrate. What is the attributions happening towards a product, which is nothing but a drug particle size distribution? It's very, very important for this lactose. What is that information we have to take in? What kind of range we have studied for this lactose? A particle size should be in between 70 to 100 micrometer. That's the study what we have done it. So what actual data for the exit batch we have observed it, which is nothing but 85 micrometer. Is this 85 micrometer between this range? Yes. So what's our recommendations for the commercial? The same recommendation has been made for the commercial. It's nothing like 70 to 100 micrometer. To ensure sufficient flowability and batch to batch consistency, these parameters should be defined. Here, there is some typographic error was observed. Excuse for me, it is not a 10 micron, it is a 100 micron. Hence, like this copy has been taken out for an example. And also, if you look at a polymorphic form can be studied, a melting point and XRBT can be used to distinguish a different polymorphic form. If you look at a melting point which is almost close level where we have observed it from range studied and exhibit batch and the proportion given to the commercial also. And the XRT pattern also have been defined based on the theta values. The same has been proposed for the commercial. So having this control strategy, you can very well optimize the formulations and provide the recommendations to the manufacturing side. What all the parameters should be controlled as a part of their operation. Similarly, find the control strategy for drug substance, excipients, process parameters, in-process testing parameters shall be evaluated. For example, for parental, you can have it microbial attributes, B, T, BAVD. For nano suspension, you can have it particle size, viscosity, and acid droplet, etc. A control strategy, here we have provide one example how that should be defined based on the manufacturing step. A suspending agent phase, what is the purpose of that? It's nothing like you need to get a complete hydration to ensure the homogeneity and uniformity of the phase. How this process parameters controlled? You can list down based on your experience or based on your few experimentation activities. Dispensing rate to avoid clumping, shear rate of the dispensing unit, mixing speed and time, and rinsing of piping. These all the process parameters should be controlled. And what kind of CQA is getting impacted? Viscosity, APA particle size, distribution, suspendability, and preservative assay. Similar way, you can also define all the manufacturing step, what all we follow, and accordingly the purpose and process parameters, you can define it, and what kind of CQA is getting impacted. Now, so far, we have discussed a basic information of QTPP, CQA, CPP, and CMA. Finally, of course, we have discussed a small introduction of control strategy also. Finally, a failure mode effects analysis, which is nothing but FMEA. As per ICH Q9 guideline, FMEA provides for an evaluation of potential failure modes for process and their likely effect on outcomes and their product performance. It is a structured approach to discovering potential failures that may exist within the design of a particular product or the process. So this will tell us on each process parameters, critical quality attributes, and your CMAs and your CPP, how it is being controlled, how it is being categorized, what is the risk factor, and what kind of actions to be done post action, what kind of level you are defining, whether the level is acceptable range or do we need to study in future, and what kind of a control strategy in place to minimize or mitigate the risk. Here we have just put one example how this is being designed and what all the studies normally they follow. Once failure modes are established, risk reductions can be used to eliminate, contain, reduce or control the potential failures. FMEA relies on product and process understanding. FMEA can be evaluated by cause and effect diagram or Ishikawa model, which is we have defined below for your example, covering all these important factors: a man, a machine, a material, a process, and your other parameters if you feel that anything method or getting impacted. So these all the assessment has to be done and find out your problems. For example, if you have a degradation issue into the product, then you can study all the factors. What are the factors involved in man, material, method, and machines? So these all factors you can study and understand where exactly your problem found. So as we discussed in previous slide, here we have listed down potential causes or root causes will be classified as material, machine, man, method, and miscellaneous. Of course, an environment. We call it 5M. 5M is material, machine, man, method, miscellaneous. So these are all the potential causes or root causes could impact your final product quality. Here is an example. How a case study has presented here. For example, for the man, handling error; for the material, API degradant, excipient degradant; And for the machine, calibration error, HPLC error. It's all just a basic information have presented here. A method is uncontrolled dissolved oxygen and uncontrolled headspace. Of course, if there is an oxygen sensitive product, could produce this kind of an unknown impurity. If the machine is doesn't calibrate it well, that would give erroneous results. For example, the HPLC is not done the calibrations properly, and it would give certain times erroneous results. And the material, the input API already having certain amount of a degradant would cause the final impact on the product quality. How this FMEA shall be defined? As we discussed initially, each risk shall be identified with the numbers. As the numbers are more, the risk is more. The numbers are less, the risk is less. That's a common understanding. But how it is being calculated? It is called risk priority number, RPN number. How it is being calculated? The RPN gives by severity index multiples with frequency index and detectability index. These are the numbers to be defined based on your understanding, probabilities and detectability and the severity. Considering all this, you can calculate the number and present it here. Maybe we can discuss with an example. So that would give a certain idea for you. What is severity index, frequency index and detectability index? Estimating severity index. The severity of the failure shall be estimated based on the effect it may have on the process, product, patient. Assign a quantitative value to the possible effect of each hazard according to scale shown in table. When estimating severity, take any defined assumption into considerations. In call cases, make a conservative estimate of severity, cautioning on the side of safety. Document the severity index value in the appropriate column of FMEA template. Let us look at how this template. This is an example. If you look at the ranking, it has been categorized from 10 to 1. 10 is, of course, we know, which is hazardous without warning, and 1 is none. When hazardous without warning, the severity of effect very high, and that needs to be monitored. It should be controlled and contained. Estimating frequency index. Estimating frequency index based on the frequency of occurrence, how many times it happens of each identified cause. Frequency may be interpreted as probability of occurrence whose quantitative value shall be assigned based on the scales shown in the table. Document the frequency index value in the appropriate column of a FMEA template. Estimating frequency index. By keeping this ranking from 10 to 1, where 10 is very high, failure is almost inevitable. You can have it by a probability. Maybe in 2 times, in 1 time it will have failure. But when we say the ranking is 1, the failure is unlikely. It can be 1 in 15 lakh also. So this way, you can calculate the ranking and put the numbers based on the occurrence. Estimating detectability. Detectability, what kind of technique you have to detect the failures. If you have a method, tool to define or understand the detectability, of course your risk is less. And if you do not have any tool to detect the failures, then the risk will be high. Assign a detectability index based on the ability to detect the event prior to or during its occurrence and thereby preventing the hazard of effect. Assign quantitative values for detectability based on the scale down in the table. Document the detectability index value in the appropriate column of a FMEA template. Here is a detectability index. If it is absolute uncertainty, the ranking would be 10. It is almost certain, it is 1. So design control will almost certainly detect a potential cause, a mechanism, and subsequent failure mode. If you do not have a method, cannot detect a potential cause or mechanisms, then the ranking would be high. If there is no control and design control, that would end up in taking the high detectability value, and this would ultimately give you the maximum RPN number of the product. This is a full template. We have defined it in a tabular form. What is being followed in pharmaceutical industry? The first column says that process activity, point of failure risk, effects of failure on the process, potential cause. S is nothing but severity and F is nothing but frequency. What kind of controls we have? If we have control in place, then you can put your detection. That the detection number shall be defined over here. For example, if your severity, you are saying that it is very high, consider is 10, and if even frequency occurrence is happens in 1 in 2, you can consider 10. If the detectability can be done for your process, then you can put the detectability is 1. So you multiply this and you will get RPN, which is 10 into 10 into 1 is equal to 100 is your RPN number. What are the mitigation or corrective actions? You can define it. And then accordingly, you can rewrite the severity, frequency detections, and then you can minimize the RPN number and what action has been taken. If it is action has been taken, make it S. If not no, then the RPN remains same. Then the risk will keep continuing for your process. So in this way, you can very well control the quality risk while formulating the formulation at development stage or even in the later stage of process development or at the scale up level. With this QBD update, we can end this model. I hope you guys have a better understanding on the QBD and what all the factors are being considered as a part of QBD. If wish, here we can summarize once again. A QBD is essential for any kind of ANDA or any kind of product development, which includes QTPP, CQA, CMA, CPP, and FMEA with a control strategy. So remaining a formulation development we can discuss in detail in the future model. Wish you good luck.